our mothers. So that's really good, at least all the effort into pregnancy, I think it gets paid out, right? So actually, we only get mitochondria from our mother's side. So this is kind of an overwhelming picture, I know. We go back to this one at the very, very last lecture, very close to Sinterklaas. Now it looks very far, right? But actually, it will go quite fast, I can tell you. And by that time, I really hope you feel good about it, because then by that time, you should know what all of this means, yeah? Of course, you have a metabolic map in A3 format. My advice is, starting from today, make a copy. Come with the copy here in the classroom, because in the coming days, we will do a lot of exercise. And this is exactly also given to you during the exam. And that's a lot of help, believe me, because you don't have to memorize cycle, and you have everything here. Yeah? But you should start to train with that. You cannot bring your own copy, but the copy will be given to you. But you can imagine that if you train already now, then it's going to be easier during the exam. And now we are going to focus actually on this part. So I will put a moment this video so I can also rest my voice a bit.
Soms is een klein gebaar genoeg om een hart sneller te laten kloppen. Nutella biscuits.
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria so cells can use it. Mitochondria consist of two membranes, an outer membrane separating it from the cytosol, and an inner membrane surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
Yeah? So I leave you now with this, and then we do a short break. We start again at 12:30, and then we continue with the other part of the lecture, yeah? Yeah, ready? Okay, let's continue from where we left, right? So we do have now energy in coenzyme that come from different reactions. We will show that at the end. We need to make ATP. How we release this energy? As I told you before, the energy from the coenzyme is carried as hydrogen form. So there are proton and there are electron, right? So what happened here, and it's important for you to understand why also NADH in general is higher than FADH2 as energy content, is that so NADH will actually release the energy at level of complex 1. So as you saw in the video, we have different complex. This is 1, 2, 3, 4, and this is called 5 or ATP synthase. These are all in the mitochondria, inner membrane, right? And it's like a family of proteins sticking together, and they carry the energy throughout the last part, where then ATP can be done. So NADH arrive at complex 1, and then it gets oxidated back, so it can be used again. And in this process, it will liberate electron and a proton, of course. Yeah? Then the electron are moved into coenzyme Q that become reduced. Coenzyme Q is actually a diffusible molecule. Yeah? and then they will travel to the complex 3, and then cytochrome C, and eventually in complex 4, they will be accepted by oxygen to make water. During all this passage, actually, the electrons slowly will lose energy, and then eventually are accepted by oxygen to make water. In the meantime, what happens at every complex they pass by, protons are actually pumped out in this intermembrane space between the two membranes of the mitochondria that I showed you before. Yeah? Now, all these protons that accumulate actually are very energetic at this moment, but it's not the proton itself. It's the fact they are accumulating in such a microenvironment that actually give what we call a proton matrix force to the ATP synthase that will use a certain amount of proton to actually make ATP. So if you think now about it, energy arrives in the form of reduced coenzyme. It releases the energy at the complex, and what happens is that the electrons are transferred eventually to oxygen, and the energy at this moment is still converted into the proton force that is accumulating in this microenvironment that can start up basically ATP synthase to work and make ATP. Yeah? The difference is NADH will release the energy at complex 1, while 5-DH2 will release the energy at complex 2. So it will skip the first complex, and this means also it will lead to less proton formation when it's about 5-DH2. Yeah? This one. Why do we say then that it's 2 and alpha ATP? Because this N proton here is not mentioned, is actually 4. So to make 1 ATP, complex 5 will need 4 proton. Yeah? If you now count NADH when it releases energy to what it leads, so you will have 4, and 4 is 8, and 2 is 10 proton. Yeah? Do you agree with me? Because it releases at complex 1. So NADH will lead to 10 proton force. 5-DH2 skip the first 4, so it's going to be 6 proton. And if you know that for every 4 proton now you make 1 ATP, now it should be clear to you why 1 NADH lead to 2 and alpha ATP, and 1 5-DH2 lead to 1 and alpha ATP. Is this clear to everybody? Why is that? Do you have any question about this part? Okay. I have a small animation of how ATP synthase work. Now just be aware of the orientation. So we have now the intramembrane space here and not there anymore. So what happens is protons are accumulating, and then this will be used to actually make active complex 5, and complex 5 will use this energy to actually make ATP. And then ATP can be used in whatever reaction we wanted in the cell. Yeah? So at the beginning, I also told you that NADH from the cytosol and NADH that is formed already in the mitochondria, they are not equivalent as energy. Why is that? Because, as I told you, if you have NADH formation in the cytosol, this NADH needs to be transported into the mitochondria. Yeah? How it's happening? Well, we have a special shuttle for this, which is called glycerol phosphate shuttle, not active in every organ, but in a lot of organs, like heart and muscle, very active organ or tissue. And how does it work? So basically, when you have NADH into the cytosol, I told you, it cannot enter into the mitochondria. This is not possible. There is not a transporter, a direct transporter for NADH. So the cells use a kind of a trick. So what they do, they oxidate NADH back. So NAD plus, again, can be used to gain other energy. This energy is temporarily transferred to the hydroxyacetone phosphate, this molecule, that is converted into glycerol triphosphate. And you can spot the 2 hydrogen are this one. Yeah? These are the hydrogen from NADH. By the way, sometimes it's written NADH, but remember it's NADH plus H plus. Yeah? There are 2 hydrogen. So the energy now, again, this lecture is about conversion of energy. It comes all from glucose, but we are moving around. We are playing around with different key molecules. This energy is now in this molecule. And you would wonder, why would I do that? Well, because the mitochondria have a specific transporter for this molecule. So now the molecule can bring this energy from the cytosol into the mitochondria. It's just a trick to bring in energy. And when it's inside, what is happening is actually the opposite reaction. So what in the cytosol lead to energy transfer from DHAP to glycerol triphosphate into the mitochondria, the opposite happen. But the problem here that you can see, and this, by the way, has also transported that can be recycled. So these molecules don't change. It's just a pool of carrier that transfer energy around. The only problem here that you can see with this specific shuttle, however, the reverse reaction in the mitochondria doesn't lead to NADH formation, but will lead to FADH2 formation. So there was a loss of energy in this transfer. And this is why I told you that the mitochondrial NADH, which is already formed inside the reaction in the mitochondria, and we will see in a moment like the Krebs cycle, it will lead to 2.5 ATP because it's already in the mitochondria, will go directly to complex 1. Yeah? However, if you have NADH in the cytosol, yeah, it needs to use the shuttle, and in shuttling, there will be some loss of energy because it's actually converted into mitochondrial FADH2. And in this case, how many ATP then we will have based on what we just discussed? 1.5. Yeah? Is this concept clear to everybody? Yeah? Why we lost energy? Yeah.
How much would the loss of energy be in space?
What do you think?
Yeah,
1 ATP. That's the difference. You lose energy. So if you do the calculation, if you know that 1 ATP is about 33, I think we say, right? Kilojoule or something, 31. That's the amount of energy that is lost in the transferring.
Yeah, but basically it's not really lost, right? Because it's only lost if you put the NADH in the mitochondria. Because there you need a transport for it.
No, if you do this, you will lose energy because you go from some. So if this NADH now in this reaction, in a different reaction, would have happened in the mitochondria, that NADH, mitochondrial NADH, would go directly to complex 1, what I show you here, right? Would go directly there and then lead to 10 proton and 2.5 ATP. But because the compartment when the reaction occurred was the cytosol and need to be transported through the glycerol triphosphate shuttle, you lose 1 ATP in the transport, in the conversion. So you actually lose 1 ATP. So there is a loss of energy in this case. Yeah? So now I don't want to make it too much complicated, but you should know that not all the tissue use this shuttle. So sometimes, I'm sorry, this is how we are made. It's not my fault. Sometimes there are tissue, and we will see later on, and it's also in your metabolic map, so you can actually find it already, in which, with a different shuttle, you can bring in the same NADH as NADH. And in that case, of course, it will be the same amount of energy. However, in the course and standard in the exam, if there are cytosolic reactions that produce NADH cytosolically, always assume that they use this shuttle unless it's stated differently. Okay? But you should know that, and we will come back to that at the end of the courses. Not now, because it would be too much. So for now, just remember this. But just for you to know, there are different shuttles. This is not the only one. Yeah, this is what I just told you. Okay? So remember, oxidative phosphorylation is made by the electron transport chain. These are these protein are called because indeed it's a chain that transport electron. Take place into the mitochondrial crystal, regenerate NADH and FADH by oxidation, and phosphorylation of ADP to ATP with the final complex. And remember, for now, that cytosolic and mitochondrial NADH only. So for FADH, there is no issue. Cytosolic FADH is the same as mitochondrial FADH. It's only for NADH. They are not equivalent because NADH cannot diffuse into the mitochondria. And it needs a shuttle that lost some energy. Yeah? We are still on board? You are still with me? Yeah? Okay. Then I want to touch upon a concept that is coupling and uncoupling. I think it's really nice and, well, I think you would be interested into it. In this case, when we couple the energy from the coenzyme to the production of ATP, we say that this is a coupled reaction. They are coupling, right? They are depending from each other. However, there is also a moment in which you can have uncoupling, and this is a very hot topic because, as usual, for loss of weight. And it's about eating, but actually try to make less ATP, because ATP is energy, and if you don't use it, it will be used to be stored or to do something else. How do we do that? So normally, and I want you to focus now first on this part of the slide, we do have in some tissue, like the brown fat adipose tissue, we do have a lot of protein called UCP. We have UCP1, 2, there are many that are called uncoupled protein. Uncoupled, as the word suggests, it's like something is getting broke, right? So what it happened, these proteins are localized in the membrane, and what they do, they actually use this proton and just let it flow inside the mitochondria. So the protons cannot be used to make ATP. And that's amazing, right? Because then, well, in a way, you are eating, but you are not making all this energy. However, this is naturally and physiologically only for certain tissue, also in certain stage of the life. And this is indeed, I mentioned the brown fat adipose tissue. And why do you think this is happening? Where does this energy then go if it goes inside? What do you think? Heat. Very good. Yeah? Babies, when they are born, even if it's summer and very high temperature, they still need a blanket for the very first few weeks because they cannot thermoregulate themselves. And they have a lot of brown adipose tissue for this reason as well. And then normally with age it declines. Chemically, there is also a lot of drug. So there is this drug like the nitrophenol, a lot of weak acid drugs like ecstasy, actually, that work exactly the same. And this is why people that are addicted to this drug, you will notice immediately they have a big loss of body weight, you can imagine, right? They dissipate all the energy actually as heat, right? And this is, of course, physiologically is not something that you would have in your body. So this is something that can be done with drugs. And I know that there are some countries in the world in which these drugs are also allowed. So but there are many that work in this way. This is just a small things I want to show you. These are images of PET, which is positron emission transmission. It's done, I don't want to go into detail, but basically these people just drink some glucose, which is, it has a tracker, and then with this special technique you can clearly see the highly metabolic part of your body. And here, basically what you can already see is that in general women has a bit more than brown adipose tissue. Apparently this has to do with the fact that we have to compensate with less in general lean mass compared to the male. Of course, if you are pregnant, these also tend to increase, or if you are lactating. So it's just an extra need for the body. But also what is nice here to see, and the pre and the post in male and female, is that they did what we call acclimatization experiment. So they exposed these healthy people to 10 days in a very cold temperature. And as you can see with the spot, they actually notice that the brown adipose tissue, it actually goes up. And especially, normally, is always the shoulder part of the body. Yeah? So I just wanted to show you because I think it's nice to see. And that, of course, you can work on it, like to have more brown adipose tissue. But this is also, in a way, why also, I don't know if it's the same for you, but in general, the same winter, you have a kind of need to eat a bit more, right? Because you need also a bit more heat to keep your body temperature. But we will have a lecture about that thermoregulation later on in the course. Yeah? So that was supposed to be a break with the question, but every year I'm late, so we do together this one. So what is true in case of uncoupling of oxidative phosphorylation? I just show you the coupling uncoupling, right? Do you think that no substrate is oxidated, no oxygen is used, no ATP is formed, or all of the above? Please don't go automatically for all of the above. Just think about it. I mean, if you want as an answer, is okay. So what is true? What is actually happening? A, B, C, or D? Who goes for A? Can you raise your hand? Who goes for B? No oxygen is used. Who goes for C? Who goes for D? No, nobody will go for D after what I say, of course. No, it's no ATPs form. Remember, we just show you. So if you go here, then you will use the proton force, actually, to make heat, and then the ATP synthase cannot use it to make ATP. But oxygen is not an issue at all. It's that the ATP is formed. Yeah?
That the ATP is formed, yeah?
The oxygen, by the way, maybe it's good to mention, is actually used before. Yeah? There was some people that say oxygen. Oxygen is used here when all the electrons are transported, but actually the proton are used independently from the oxygen at the end.
From the oxygen at the end, yeah?
So isolated mitochondria, we can isolate it in the lab. We break down the cells and we just isolated mitochondria. If they are incubated with 10 mole of cytoplasmic NADH, yeah? How many mole of ATP now will be formed? Now I want a good answer. I just explained this concept. So we have just mitochondria, and we put outside the mitochondria 10 mole of cytoplasmic NADH. How many ATP we can do?
We can do?
Yes, you have the good answer. 15. Yeah? Because we say 1 mole of NADH from the cytosol will lead to 15. And if it was mitochondria, how much would have been? Anybody? Give me a number, a good number. Huh? Yes. Yeah? Because that would have been already in the mitochondria. In that case, it's 1 NADH, 2.5. Yeah? And what if you use a homogenate of mitochondria? So if we take the mitochondria and we do like a pesto, what do you think it will happen? Anybody? We don't make pasta for it? With it, of course. Yes?
I would say ATP to some extent, but if it's uncoupled, like you don't have the chain
Yeah.
that is so continuous, so it would be
You're really right.
I don't know. I mean, some energy would be but it would be less.
Yeah, you're really right. Yeah, that's correct. Actually, there is nothing happening. So you really need, and we will see this at the end of the course, you really need actually the complex to be in a certain order and next to each other. Yeah? And the structure of the mitochondria should be intact. So it's not about coupling or uncoupling, but should be intact so that the complex can work. So in this case, there is no formation, and I have the answer here. Yeah? So that when you watch the recording, hopefully, maybe, and otherwise from last year, then you have the answer. Yeah?